Hello friends, Basis-Botany, next episode. In this episode, we'll be discussing molecular basis of inheritance, sixth part. And what all things we'll be discussing in this episode. For that, I will share the screen, so it will be convenient for you. I think screen came. So we can proceed. So molecular basis of inheritance, sixth one. And what all things we'll be discussing in this episode? As usual, one introduction, then transcription in prokaryotes, transcription in eukaryotes, followed by 5 theory question, 5 NEET model question as usual with the key. So we can proceed with the introduction. So generally the term transcription. Transcription generally means change into a readable form. Wherever we use the term transcription. Here in genetics, molecular basis of inheritance, transcription basically means production of RNA. And that is usually from DNA. Transcription can be seen in both eukaryotes as well as prokaryotes. And the main enzyme involved is DNA-dependent RNA polymerase. So we'll be discussing the transcription part mainly in this episode. So we can enter into the topic. So first we have to start with the transcription in prokaryotes. Prokaryotes here mentioning about bacteria. As you know, bacteria, no nucleus. So no well-defined nucleus. So the events connected with the transcription more towards cytoplasm. Enzyme is DNA-dependent RNA polymerase. And all the 3 RNA will be involved. And only one type of RNA polymerase is involved. So 3 RNAs will be there, only one type of RNA polymerase. And that RNA polymerase can transcribe all types of RNA. Typical example, everything I written here, so it will be convenient for you to refer. mRNA provides the templates. tRNA will bring the amino acid and reads the genetic code. While rRNA, it plays structural and catalytic role during translation. This is what given in NCERT page number 93 is important one theory, fill in the blank and need to match the following type question. This diagram, beautiful color diagram connected with NCERT. So everything mentioned here. So before getting into it, I will explain certain basic aspect and we can refer this. Hoping that first page is clear to all of you. So we can proceed to second page. Here everything, I told you everything I represented, this will be easy for you to refer. So first we'll start with the enzyme. RNA polymerase is the enzyme which is a holoenzyme, consists of 5 polypeptide chains. They are 2 alpha subunits, 1 beta subunit, 1 beta prime subunit, and a small omega subunit. So these 5 polypeptide together called core enzyme binds with a sigma factor to form a holoenzyme. Holo means complete enzyme. So RNA polymerase, holoenzyme, which consists of basically 5 polypeptide chain, 2 alpha, then 1 beta prime, and a small omega subunit. So these 5 together called core enzyme binds with a sigma factor to form a holoenzyme. I think that enzyme naming clear to all of you. I represented here. So this part is called core enzyme without sigma unit. If sigma unit is added, then it becomes holoenzyme. Now, transcription is completed in 3 steps. One is initiation, second is elongation, third is termination. That is the usual procedure. Head, body, tail, that way. So here it is 3 steps: initiation, elongation, termination. Now comes to initiation procedure. So starting, it is catalyzed by the initiation factor or sigma factor. RNA polymerase binds to the promoter. Promoter means P site normally where polymerase will bind. Operon also same thing you can find. Initiate the transcription. So when sigma factor attached to the promoter, transcription begins. And for this substrate used is nucleoside triphosphate and polymerase in a template-dependent fashion, following rule of complementarity, NCERT, important for exam, just like DNA duplication. So substrate used is nucleoside triphosphate and polymerases in a template-dependent fashion, following the rule of complementarity. That is why I told everything I written there with the different colors. In the absence of sigma factor, transcription can start, but non-specifically by the core enzyme at any base of DNA. That is regarding sigma factor present and absent. I think it is clear to all of you. Now, it somehow also facilitate, it means RNA polymerase, opening the helix. After elongation process, these all NCERT lines and important for theory entrance exam. Now, elongation. Elongation is facilitated or catalyzed by RNA polymerase core enzyme. And that can only do it, only capable. Now comes last part of it, it is called termination. Termination means once the polymerase reaches the terminator region, the nascent RNA falls off. So also the RNA polymerase. This even lead to the termination of transcription. And for this purpose, rho factor, here you can find the emblem. Rho factor is required for termination of transcription. This is how the three steps: initiation, elongation, and termination. Now, here I wrote a small line. Only or a short stretch of RNA remains bound to the enzyme. And this all line by line, when they ask for NEET or a theory exam, it will be confusing for us. That is why I put that line there. Line number 94, reprint edition, March 2024. I think this page also clear to all of you. Now we can proceed to next page. Okay. So in transcription, here you can find transcription, RNA polymerase somehow also facilitates opening of helix and continuous elongation. So in transcription, RNA polymerase, not helicase or anything, RNA polymerase somehow also facilitate opening of helix and continuous elongation. Here you can find. Again, that is the line in NCERT page number 94, 24 edition, March, reprints. But when we come to a comparison between eukaryote and prokaryote, in comparison with the eukaryote, here you can find, there are certain variations in bacterial transcription because bacteria is a prokaryote. So one, mRNA can be active without any processing. Processes will be studying eukaryote. Two, in prokaryote, there is no much separation between cytosol and nucleus since they are in same compartment, no separate nucleus. How it affects? Its effect is transcription and translation can take place in same compartment. Third one, before the completion of complete transcription, translation can begin many times. That means transcription and translation can be coupled in bacteria. Theory question, NEET model question. I think prokaryote transcription is clear to all of you. Now we can proceed to transcription in eukaryotes. In eukaryote, two additional complexities are there. Again, theory entrance question. One, there are at least 3 RNA polymerase in nucleus, in addition to the RNA polymerase found in organelles. Then for these 3 RNA polymerase, there is a clear-cut division of labor. Again, that is another important question here you can find. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Now, RNA polymerase 1 transcribes ribosomal RNAs with the 28S, 18S, 5.8S. Connected with the RNA synthesis, this part is important for theory NEET. RNA polymerase 2 transcribes mRNA, otherwise called hnRNA, heterogeneous nuclear RNA. Then RNA polymerase 3 transcribes tRNAs, otherwise 5srRNA, snRNA, small nuclear RNAs, or scRNA, small cytoplasmic RNA. So that is why the clear division of labor in eukaryotes. I think that page is clear. Now we can proceed to next area. So new or nascent RNA synthesized by RNA polymerase II, that is mRNA, here we can find, is called as hnRNA or heterogeneous nuclear RNA or primary transcript. Important. It has to be modified because it contains useful part and unwanted parts or bases. So primary transcript has to be modified. I think up to this it is clear. Now we can proceed to next page. Here you can find. These all are beautiful diagrams clearly explaining everything labeled there. Once it asks for NEET exam. It's a theory question also. All process I will explain, so no need to be get tensed. Okay. Now processing of primary transcript. So non-functional base sequence is called introns. Functional base sequences is called exons. So introns and exons. Naturally, these two has to be separated. Similarly, to be functional, many other events has to take place. I think we can proceed to next area. Post-transcriptional procedures important. That is converting primary transcript to functional one. Mainly 3 steps involved. One is capping, tailing, splicing. Capping, tailing, splicing. CTS. So when we write it in a serial manner transcription, then capping and tailing, splicing, this way probably procedure may be going. Here we can find written there. So transcription, capping, tailing, transcription entire procedure, capping, tailing, splicing. I think it is clear to all of you. Now we can proceed to next page. Now each item I explain what is importance, what is the need, which all chemicals, including diagram. Since notice the diagram, you can easily follow. First, capping. Here also you can find capping. Below it I wrote the function. Before it we can proceed here. Capping will be at 5' and what is meant by it, what is the usage of it, etc. What is the chemical? See, rapidly after the start of transcription, an unusual nucleotide, which is called methyl guanosine triphosphate. Normally it is called 7-MG, etc. Methyl guanosine triphosphate is added at the 5' end of the primary transcript. That is, I told you, diagram clear, that much explanation I given there. So 5' end of the primary transcript, this methyl guanosine triphosphate will be added. Enzyme is guanylyl transferase. Guanylyl transferase. Guanylyl transferase. That is the enzyme. Now, one main purpose to form mRNA ribosome complex. One main function. Now, I think that idea is clear. Cap is identified by 18 srRNA of ribosome. Last page we studied RNA polymerase. Or translation issues can be possible. I think this part is clear to all of you. Now here, capping. Different functions will be there for capping. One is protection, since it comes to the cytoplasm, translation, efficiency, specific other functions, identifications, and looping. All can be there. Now coming after capping, next part is tailing. Tailing here you can find, why I'm telling tailing in between, after the finishing tailing will be discussed. Tailing. It is concerned with the tailing. Adenylate residues 200 to 300 are added at 3' end in a template independent manner, independent, with the help of polyadenine polymerase. That is the enzyme. 200 to 300 adenylate residue, 3' end. Here you can find tail. Here it is cap. And what is the function of tailing? It is stabilize mRNA. So covering. Now comes in between procedure, capping and tailing. So order-wise, we can find capping, tailing, splicing. This way it proceed. Now next item is splicing for us. So what is meant by splicing? In splicing, it means splitting and joining in primary transcript. Splitting and joining in primary transcript. Here, non-functional introns will be removed and functional exons will be put together to form functional RNA. Each intron starts with the dinucleotide GU and ends with the dinucleotide AG. This principle is called GU-AG rule. A complex called spliceosome is formed between 5' and GU and 3' and AG of introns. ATP can provide the energy. Introns will be removed. Adjacent exons are brought together. The ends are sealed by RNA ligase. Introns will be removed by small nuclear RNA, which is otherwise called snRNA. And protein complex called small nuclear ribonucleoproteins or snRNPs, otherwise called SNRPs. Here you can find what I just told. So splicing, everything mentioned here, including enzyme, all aspects. I think it is clear. Diagram also, self-explainable diagram, everything I labeled there. NCERT also labeled it, so it will be useful for us. Self-explanatory diagrams. We can proceed to next page. This is another diagram. May not be in NCERT, present NCERT, but old NCERT. That is why I put in inverted comma here. NCERT, old NCERT. So here everything mentioned what is splicing. So fully mature RNA, now the fully processed HRNA, here you can find, become mRNA, is transported from nucleus for translation. So these all are nucleus now coming out for translation. That is second part of it. Next episode, we may discuss it. And this is the diagram showing it. Self-explanatory, clear diagram. Primary transcript, introns, exon. Then how cap and tailing takes place. Then splicing, spliceosome, and fully mature RNA. I think it is clear, beautiful diagram, self-explanatory diagram. Now we can proceed to next page. So normally a question comes in the mind. Why post-transcriptional modification in eukaryote required? What is the use? Several reasons are suggested. Some of them include to remove non-coding regions for protection of mRNA, aid in transport, connected with the gene expression, etc. So significance of such complexity is now beginning to be understood. Split gene arrangement represents probably an ancient feature of genome. Presence of introns is reminiscent, that means aware of antiquity, and the process of splicing represents the dominance of RNA world. In recent time, the understanding of RNA and RNA-dependent process are living system have assumed more importance. This is a line or a paragraph given in NCERT page number 95, reprint edition March 24. It's an important statement, RNA world importance. After that, as usual, 5 theory question, 5 NEET model question with the key. So these are the items we'll be discussing in today's episode. So it is a little bit tougher for most of the students, but I written here, I shown here everything what is possible connected with the NCERT. So what all things we discussed in this area? One is an introduction. Second is transcription in prokaryotes. Third is transcription in eukaryote. Then 5 theory, 5 NEET model question. Transcription prokaryote, you can find which chemical, which enzyme, where it takes place, etc. And single RNA polymerase. Then when we come to next area, that is transcription again continuing. When we come to next area, we can find after initial elongation termination, transcription in eukaryotes. That is enzyme, coenzyme, holoenzyme, etc. Then initiation, elongation, termination of factor needed. Then fitting, opening of helix, that is RNA polymerase. That all written there. Next, transcription in eukaryotes. It is more complex. Nucleus is there. And 3 RNA polymerase 1, 2, 3, what the function they are doing and what happens to primary transcript, why it is needed. Then one beautiful diagram to show how the splicing with the 5', 3', capping, tailing, all this with introns, exons, all this has been shown. Then after this, how it will be sealed, RNA ligase. Then another beautiful diagram, present NCERT, not contains it, old NCERT, everything mentioned here. I think it is clear. Then why post-transcriptional changes needed in eukaryote, then significance of such complexities, then 5 theory question, 5 NEET model question with the key here. So if you're able to follow this, if you like it, subscribe or like it, share it, comment on it. We can meet with the next topic in next episode. So thank you. Take care. Bye.